Hello class, I hope everyone's doing well when they start watching this video. Coming up here, not this week, but the next week, we're going to be doing a science day at the school. We normally would be doing science fair, but just with the way this year is and all the challenges and circumstances that we find ourselves in, we're unable to do a normal science fair, sadly. So instead we're going to do kind of like a science day. But to get us kind of thinking about science maybe in a different way or in a new way, we're going to be reviewing the scientific method with some fun, I think just one fun example, great video about what's called an iodine clock. So we've been learning about different reactions, chemical reactions, so I thought it would be kind of fun to do a very chemistry-based experiment as we review the scientific method by doing that. So, as always, I like to ask us questions to get our brains thinking, so just start to think about what do you guys already know about the scientific method. I know you guys have done science fair before, so what do you already know? Also, what are some important steps? So, do you remember any of the steps that we used in the past for the scientific method? And then also, I didn't put this on there, but what are some experiments you have done before? What were your results? How did they turn out? I'm curious to hear about those as well. So our objectives, we want to review the steps of the scientific method and then kind of brainstorm together and watch a really cool experiment. So our steps in the scientific method, first is to make an observation. Our circle over here, I think, does a great job of illustrating how it works. So our arrow is starting with observation, and then it goes to question. So we want to ask a question after we have an observation. Then we want to test our test a hypothesis. So we'll come up with a hypothesis, and we want to test it by designing and doing an experiment. And then we want to have an analysis of our experiment. What happened? What did we notice? And then a conclusion. So those are the steps. I really like how this is in a circle though, because a lot of the times we end with a conclusion and then have more questions or more observations that we didn't expect. So maybe we run through our experiment, we have an analysis and notice that something's different than what we thought it would be. So we want to make another observation and another question, another hypothesis, and it can keep going over and over and over. So what we're going to do is watch this really cool video. It's called an iodine clock reaction. And I am curious what you guys notice in it. So we're going to start by doing this, and then we're going to make an observation about what happens. I'm going to link this video, as always, in our classroom. So please pause this now and go watch this video, and we'll continue in this presentation. But a fun fact is this is the exact experiment that made me want to study some more chemistry in college. So it's really cool. I hope you guys enjoy it as well. But first, our observation, what did we see in that video? Well, I noticed that what happened is we have a very clear liquid go to a dark liquid very rapidly. So we notice here this picture, we start with a clear liquid and we go to a dark liquid. I'm curious if you guys notice anything else, maybe how long it took, maybe how many times they switched it back and forth, anything else, maybe what they used in it, any other observation. But we're curious why. So we have this observation, but we have a question. Why? Why does this happen? Well, we can use our scientific method to learn about it. So some possible questions. I'm curious what you have to say or what you have to think about after the video, but some things that I have is, why does this happen? A great question to start with. Also, I'm curious is, would increasing the concentration of one of the liquids speed up the reaction? So if we made it really, really strong, would it make it go faster, slower, or not at all? Would it not change at all? I'm also curious if we made it hotter. What if we changed the temperature of the liquids? How would that affect our experiment, our reaction? So we're going to stick with, we're first going to now do a hypothesis or educated guess. A lot of times this requires more research. In this example, more research about what is called kinetics, or the speed of reactions. But the question I was hoping for us to look at more is the concentration. How does the concentration affect things? So I came up with the hypothesis that by increasing the concentration of a liquid, the reaction will speed up. So that is my hypothesis, or our hypothesis, for us to test. So how can we test this? Before we get into this, I want to make a quick note on our variables, because we want to be able to limit our variables just to one variable that's changing so that we can get better results. So for example, just to review, we have our three types of variables. We have a dependent, independent, and a control variable. So our dependent variable is a variable we can measure. Our independent variable is a variable that you can control, and a control variable is a variable that you keep the same for all conditions. So we have a nice little picture up here that does a good example of showing this. This person is trying to see how the time of day affects how much fish he will get. So if fishing in the morning he gets more fish than at maybe lunchtime when the fish are hungry, maybe dinner time when if a fish hasn't had food all day, then maybe they're really hungry at dinner time. So he's curious how the time of day, his independent variable, the one that he's going to change, will affect how much fish he can get, which is the dependent variable. But as he's doing this, as he's changing the time of day, he wants to make sure he's keeping things like the type of bait and the type of fishing pole he's using constant. Because let's say he changed the type of bait. Let's say in the morning he used worms, but in dinner time he used hot dogs. And he caught a lot more fish in the morning. He couldn't for sure say that it was because during the morning he caught more. Maybe fish only like worms and they don't like hot dogs. So we're confused on which variable caused the fish to bite more. So we want to keep the type of bait the same or the type of fishing pole the same, or our control variables. But going back to our experiment, we want to make two different solutions with different concentrations of our iodine is what we're going to try to do. We want to be able to mix them in the same solution in different beakers. So we're going to have, the way this works is we take our solution with iodine and then another solution, working solution. So we're going to have the same working solution, but we want different concentrations, a stronger iodine and a weaker iodine solution. And then we want to time to see how long it'll take the solutions to change colors. So, for example, our dependent variable in this case would be our speed of the reaction. That's what we're curious about. That's what we're searching for. Just like we were searching to see how many fish we could get, we're searching to see if we're going to speed up the reaction, slow down the reaction, or our reaction will not change at all. Our independent variable is going to be the concentration. So this is the variable we get to change. So we're going to test it once with a normal concentration and once with a really strong concentration. And then our control variables are our amount of the solution, our size of the beaker, temperature, if we're stirring the mixture or not. So everything else that could possibly have a impact on our reaction, we want to keep the same. So those are going to be our control variables. So here's our video of someone doing the same reaction that we saw in the very first one with different concentrations. So I'll have that linked in our classroom in the same place as always. So I'm curious in our analysis is what did we notice? What did we see with our eyes? So what I noticed is the reaction with a higher concentration occurred a lot faster, but I want us to use better, more descriptive things. So if we could, we would have a timer next to it to get concrete or really good measurements, but sometimes all we have is what we see. So that means we want to be very descriptive in the words we use to explain it. So I thought what I saw was a fast and sudden change that takes only a couple of seconds in our strong concentration, while it took about 10 seconds in our weaker concentration. So those were how I described it. Our conclusion, we want to answer a few questions. The first one is, was our hypothesis correct or not, and why? So yes, our hypothesis was correct because the liquid with the higher concentration changed colors faster. And then what could we improve about the experiment? Some other things we could have tested is we could change the temperature of the solutions or test different amounts of iodine. So rather than just concentration 1 and concentration 2, maybe we had 5 different concentrations. Maybe some temperatures were hotter than others to see how that worked as well. And then I'm curious, is after watching this, if you guys have any other questions relating to this reaction or this experiment. So we always want to end with, I'm curious about, I don't know, why is iodine this color? You know, why does it change to be the color it does? Why does it not change to be red, for example? So we could test more and more things. This video that I'm going to post actually explains in a lot of detail how this works. So if you guys are interested, wanting to know just as what is exactly is going on here, this video is about 10 minutes, a little bit longer, but it does a really good job of explaining everything that's going on. So that's all I have for you guys. As always, if you have any questions, please let me know. I hope you really enjoyed this video. Like I said, this is one of my favorite reactions. I wish I could do it with you guys, but we don't quite have the materials to do it. But it's a really cool one that I hope you guys like.